Okay, we all set? All good? Okay. Okay, so the final talk in this session is on adaptive management, and I'm going to kick on straight from Zoe's talk and use the excuse me, just got to get my screen set up here. The reintroduction of Totowa to Totowa as a case study. And I guess the starting point is this introductory slide is showing the release, which was the moment in April 2021 where we released the Totowa. But of course, as Zoe has explained, that's not where the project ends. And of course, it's also not where the project begins because a whole lot of planning and thinking went in beforehand. But where I want to go now is to make the point that there is a much longer and broader context around a reintroduction like this one. And if we're talking about mainland bird reintroductions in Aotearoa, I think we really need to go back to well, possibly the late 80s, but then at least to the mid 90s, where the mainland island program started up. And initially there were those 6 sort of dock mainland islands here shown on the left. And of course that inspired this great proliferation of projects that we have now. And a major theme, of course, of these projects has been reducing or eradicating invasive predators, but also bringing back native species. So in theory, because there's all this work that's going on, we should be able to progressively learn about what we're doing in terms of reintroductions and be able to make better and better decisions as we're able to make better predictions. So the theme of this talk is really to talk about how this process of adaptive management can work. For those who are not familiar with adaptive management, what it should mean is not that you're just flying by the seat of your pants, but that you're following a structured process for improving your decisions over time. So it's a subset of structured decision making. Now, I'm not going to talk about structured decision making in detail, but it basically involves formalizing your objectives and your management alternatives, having specific models in mind, which you use predictions, and then you use those to make look at the trade-offs and make your best possible decisions. The thing that characterizes adaptive management is when you're making not just one decision, but several decisions where you start going around in this loop. And the point that separates that is the fact that you're doing some monitoring and then you're updating your models and then able to make better predictions. So how does this updating work? It works through a process whether people realize it or not called Bayesian inference. And so I'll take you through this a little bit now. From my viewpoint, I guess reintroduction biology is basically an inherently Bayesian process. And what I mean by that is, first of all, we consider a new reintroduction you're planning. So within the context of Aotearoa, you'd be doing a translocation proposal and producing a document in this particular form. And of course, to make a proposal, you need to know something. So at that point, you gather all your information together, and that's what we call our prior information. And you use that to try to make some predictions, and amongst other things, decide whether we want to proceed to a reintroduction. Okay. If you do proceed, you release the animals, and hopefully some monitoring takes place. And at that point, you actually get some site-specific data. Okay, so we didn't actually have any data from the site before because there weren't any birds or whatever in it. But now we've got site-specific data, and what we're now able to do is add that to what we knew beforehand, and we're able to make what we call posterior predictions. And we're able to do 2 things. One is adjust our management, for example, the level of pest control at the site. But the other thing is when the next reintroduction or translocation proposal comes along, we should then be able to update our prior information and be able to make better decisions. Okay, so where does the Bayesian come through? It's because Reverend Bayes, Thomas Bayes in the late 1700s, worked out what the maths were for this process of combining our prior information with new data. And of course, now we have lots of software that we can analyze all sorts of complex data with. So how does this actually work? Okay, so I'll take you through the example of, well, Turitea Totowa reintroduction, but also Totowa in general on the mainland. So what you want to start with is actually considering of what your initial model is. So you basically start with something we call a conceptual model. And with populations, what we typically do is you might come up with a diagram like this one. It says adult females, well, they produce babies, they might become juveniles, they'll recruit in the population, those adults have to survive, etc. And then what are the limiting factors on those vital rates that might be affected by our management? And with Totowa, what we knew a priori was that the reproductive success was going to be quite strongly affected by predators, particularly ship rats. We also knew that some of those females are probably going to be killed on the nest. So depending on how many ship rats are out there, that's going to affect adult female survival. The other thing we guessed was that because juveniles undergo a dispersal phase in sites with high connectivity, as Zoe's going to talk about, some of those are going to leave, so that's going to affect your recruitment, and they might be affected by predators as well. So then what we want to do is actually collect data and parameterize these different rates. So with Totowa reintroductions on the mainland, this really started in 1999 when we released them to Pangaroa mainland island, which is one of the original 6 mainland islands near Taihape. So that's what the bush looks like there, or part of it. And here's our cast of characters. We've got our reintroduced Totowa. Hopefully they're going to produce things like this, which are baby Totowa nests. These are negatively affected by these things, ship rats. So consequently, we try to manage these things, in this case, by feeding them Brodifocom and manage and monitor the success of that, in this case, using old-fashioned tracking tunnels. And Pangaroa was an interesting site. If you just want to look at this bottom graph here, we've got rat tracking rate over time from 1999 up to 2004. What's going on here is you can see it started somewhere between 20 and 40% rat tracking. And as a result of monitoring the robins, there was more effort put into that initially till it got down to quite low levels down here, about 5%. And then, if you remember, there was a national policy decision by DOC in 2002 to stop using anticoagulants on the mainland except in emergency situations. And so basically the predator control stopped, and then the rat tracking went through the roof, which was not good for the Totowa, but it did give us a very good start on working out how vital rates responded to rat tracking. Or rats as measured by rat tracking. So in this graph, you can see, in this case, what we've shown is lambda, again, the finite rate of increase. Remember, lambda needs to be greater than 1 for a population to grow. Here's our rat tracking rate here on the x-axis, and basically, and the dotted line showing the amount of scattered, and we've got some alternative models there, but overall showing predicting this rat tracking rate needed to be about below about 20% or so for a Totowa population to persist at Pangaroa. That, however, is just one site. So what we then want to do, as more reintroductions took place, we've got more people collected more monitoring data, and we've slowly built that up. So going forward about 10 years, this is now showing a paper that came out of Liz Parlato's PhD thesis, where she put together the data for 10 different sites of all Totowa reintroductions, all the vital rate data that were collected, the rat tracking date, the connectivity indices for all these sites, and put that all together. And putting it all together, what that allows you to do, amongst other things, is you can get a handle on just the random site-to-site variation. And what that allows us to do, if you look at this graph, you can see, again, we're looking at finite rate of increase. And again, lambda equals 1. And what Liz has done here is she's come up with predictions about what lambda will be under different levels of rat control for these existing sites, but has then done a new thing of coming up with a prediction for a new site simply based on the rat tracking data for that site and its connectivity. In that case, it was Pukaha, but here we're going to talk about Turitea. So again, you've already seen Turitea, 2,000 hectares, and on average has a rat tracking rate of about 19%. So based on what I just showed you beforehand, we're able to make prior predictions about all the vital rates based on the characteristics of the site, and produced a graph like this. This is showing a distribution for lambda, and what that dotted line placed there at 1 is showing our best prediction was that the population would increase, but the fact that the curve is overlapping is showing there's quite a bit of uncertainty about this. And this is showing population projections after reintroduction, again, before it was done, with a dotted line showing uncertainty. So this is what went into the translocation proposal. The people approving it decided, yes, this was worth a crack, despite all the uncertainty. And so we went ahead, and as Zoe just showed you, we got this nice little set of about 18 Totowa that established conveniently in this area here, which is pretty well spot on what we predicted a priori, that about 18 out of 40 would actually establish by the next breeding season. And this is now, we've got Kara here doing monitoring. So the next thing I want to talk to you about is the updating. So monitoring involves mostly Kara going out and looking at all the places we found the pairs. We've got a nice little pair here. There's a male here, female here with a bird patch. Follow the female back to their nest, then we know where the nests are, put some flagging tape on them, GPS mark them, and then follow them. And then if they manage to have young that fledge, then we catch the fledglings and band them. And this is the sort of data that we get. This is individuals shown here, and this is basically dates where checks occurred. One showed where an individual was detected as 0, where it wasn't detected on a check. Over on the right here, we can see numbers of fledged by different females. And 2 points to note: one, that a lot of zeros here. The nest success was not particularly flash. And what all these highlighted yellow bits here is we actually had a lot of birds disappear. So that initial monitoring was not particularly good news for the population. But for the purposes of this talk, what I want to show is how you can do this Bayesian updating process. I'm not going to, of course, go through the details, but here we have our different rates: adult female survival, male annual survival, fledglings per female, juvenile survival. And what we're showing here is then a distribution which shows our knowledge about these things. So this is basically our prior belief based on other sites about what would happen on Turitea for adult female survival. This is what we'd infer just based on the new data, which you can see that survival is a lot lower. We had quite a few disappearances. But that's from a small sample size. So how do you put those things together? Again, we can use the Bayes theorem, and then what we get is a posterior distribution, which suggests, okay, its survival is probably a little bit lower than we thought it was. We can do the same thing for the reproduction. We can't yet do that for juvenile survival because we haven't been able to observe that, because this is only 4 months of data. Okay. What does this then mean in terms of management? So now what we're able to do is present the stakeholders involved with the reserve, which is the iwi and the council and other people, with updated projections. So this is what we projected up a priori. What we're now projecting is that on average, we're actually looking at the population actually declining to extinction, but there's uncertainty about that. That raises questions like, okay, should we actually invest in trying to get those rats down? And this is our current projection. If we get rats down to 5%, very up in the air whether that will allow us to have a population or not. So the upshot of all this is, first of all, just reiterate the point that while releases kind of have great optics, reintroductions are very complex endeavors. They involve difficult decisions. So what we're trying to do is improve our methods for doing that. And what I've tried to illustrate here is we do now have quite good methods for making data evidence-based predictions for adaptive management. The final thing which people may not realize is when you do all this, rather than eliminating uncertainty, it often basically reveals that there's more uncertainty than we realize there is. Okay, so that's the end of my talk. I'll leave you to read what Socrates has to say about uncertainty at the bottom.
Thank you, Doug. That is brilliant. Yeah, very, very interesting to see that process and actually being taken through that translocation and how it's being followed right at the moment. At the moment, there's no questions yet for you. So again, I'm going to jump in. And I was wondering, with those site-specific variables, is there anything that jumps out in terms of, I don't know, forest structure or type or anything that you can sort of be like, oh, we know that certain sites, I don't know, if they're further south, they will do worse than if it's a further north reintroduction site?
Yeah, frustratingly, the answer is mostly no. is a little bit all over the place. The one thing I think is probably worth revisiting is that we initially thought there was a peninsula effect going on and that peninsular sites were doing quite badly, and we thought what might be happening there is that juveniles were basically funneling out of the peninsulas. And then I had a student did a master's on that, and basically this was at Tawharanui, and she basically found that there was basically no evidence for that whatsoever, and basically that it was just juveniles were dying. So we now think, oh, maybe these sort of coastal peninsular sites, maybe they might not be doing so well. So that's one possibility.
And I guess with the juveniles dying, it's probably, as always, a little bit hard to know if it's disease or if it's just not enough food or what exactly there is going on. No, that's it's certainly complex, very complex. There's one question from the audience. Doug, why did the Turritia translocated population have such a grim projection even under a 5% rat tracking scenario?
Okay, I guess the main answer for that is based on our available evidence from the different populations. And this is pretty complicated. But the difference between 5% tracking and 19% tracking, which is about where we are at the moment, is actually not huge in terms of evidence what effect that makes. So, for example, if you look at Paengaroa as an example, we're actually going basically almost from 0 right up to like 90% tracking. And we get to those really high levels, 60, 70, 80, 90% tracking, we can really see it's making a huge difference. But it's just that 5 to 20, it's, as based on our current knowledge, isn't a huge difference.
Yeah, that makes sense. Cool. Well, I will leave it at that.